Thank you, everyone. Without delay, we will start with the first speaker of the day. So it's a real pleasure to welcome Eduard Porta from the Josep Carreras Leukemia Research Institute in Barcelona. So Dr. Eduard Porta obtained his PhD in biomedicine in 2013 from the University of Barcelona. He then spent 4 years as a postdoctoral fellow in the United States at the Stanford Burnham Prebys Medical Discovery Institute, where he worked on how to use protein structures to data to identify cancer-driven genes and drug biomarkers. So between 2016 and 18, he co-led 2 analysis working groups from the Cancer Genome Atlas project on cancer-driven genes and oncogenic mechanism, and contribute to a third on the immune landscape of cancer. Then he was junior leader fellow at the Barcelona Supercomputing Center, where he worked on how germline variants shape the immune response against tumors and on the use of polygenic scores to predict cancer risk. Since 2 years, he leads the Cancer Immunogenomics Laboratory at the Josep Carreras Leukemia Research Institute. His group uses computational approaches to understand how genetic variants, both germline and somatic, interact with the tumor microenvironments, particularly with the immune cells. So the floor is yours.
Okay, thank you. So, as they mentioned, I'm Eduard Porta. I work at the Josep Carreras Leukemia Research Institute, and I'm going to be talking about understanding oncogenesis across biological scales. But before that, since this is a PhD program or meeting, I thought that it would be, it would be a good idea to talk a little bit about, one's career, so that people know that, how a career in science can look like. So I started working in industrial biotechnology with E. coli. I really didn't like it. So for my master's, I moved to another wet lab to work on immunology, specifically in gene variants related to autoimmunity. I did a lot of work with thymus samples, did a lot of PCRs, didn't like it either, and wasn't really good at it. So for my PhD, I finally found something that I liked, and I was a little bit better at. So I did my PhD in bioinformatics. All of this was in Barcelona. And then after my PhD in 2013, we moved to San Diego, where I was very lucky to join the Cancer Genome Atlas project. It ended up with 3 very nice papers. For my second postdoc, another 3 years, as I mentioned, okay, yes, that would be very nice. That's better. Thank you. So during my second postdoc, I worked on germline genetics, again, related to cancer, and a little bit about machine learning, because, it was a supercomputing center, so I had to learn about machine learning. That's also where I met Vera. And then, and then now I'm leading my own group since 2020 at the Josep Carreras Leukemia Research Institute. So, and one key advice that I would give to young PhD students would be to disseminate your work, because I actually got my TCGA papers through Twitter. So back in 2015, I wanted to learn how to use Shiny in R, and this guy, Yasin Sembavaoglu, very nice MD fellow working on some projects on TCGA and kidney, he published this preprint on the immune landscape of the kidney cancer or something. And I said, I downloaded his data, and I built this very dumb and very simple Shiny app. And I sent it to him. I said, look, I did this, if you want to take a look at it, go take a look. And, and he kind of liked it. And then he invited us to join a meeting in Houston where all the TCGA people were meeting. And then, since we went there, they invited us to join the driver paper and the perspective paper and all that, and all of those papers, which are arguably the 3 biggest papers of my career so far, so which I got through Twitter. So, disseminate your work, share it with the world, and don't be scared of that. And another more recent example, we're working on AlphaFold. This is last year, July last year. Google released the AlphaFold predictions for all human proteins. I was, for various reasons, in a very good niche spot to exploit that data. And, Pedro Beltrao from EBI, he sent this tweet, look, I've seen on Twitter that there's a lot of people already analyzing data from AlphaFold and all these predictions and whatnot. So let's write a paper together. And, and then Alfonso and I said, okay, sure, let's join. And this was, we put this paper together, I think, in like 3 months. It's a very interesting paper. We have 9 co-first authors and 9 co-last authors, because the whole idea was that, each group would bring in their own expertise. And it's about to be published in Nature Structural and Molecular Biology. So again, disseminate your work and share it with the world, because, usually good things happen. So where are we? Our group is a little bit special. We also have a very nice building, just like yours. We are mainly based at the Josep Carreras Leukemia Research Institute, but we also have an affiliation with the Supercomputing Center, so we have access to the supercomputer and all of these things. And we are 7 people. We have a wet lab manager, Daniela. She's here in case you want to discuss any spatial transcriptomic stuff. And 4 PhD students and 2 bioinformatics technicians. So we work on cancer genomics, structural biology, tumor immunology, and spatial transcriptomics since a couple of years ago when we started with wet lab. 2 job alerts. I'm going to be looking for a postdoc to work on federated learning and genomics data starting September next year. And we just got this co-fund from the European Union at the Leukemia Institute. We will be having 16 3-year postdoctoral fellowships. These are competitive, but it's small competition because it's 40 people. So if you're finishing your PhD and want to move to Barcelona in September, we will be opening this call in January 2023. You can apply with anybody, but I prefer if you apply with me. Then, last advertisement. We are coordinating a special issue. I've seen a lot of talks about spatial. So we are coordinating a special issue about spatial transcriptomics in a very nice journal. So get in touch, send me an email or a tweet or whatever, and so that we can discuss this. Now, how do we work? We have a motto in the lab, our sort of slogan, which is embrace complexity. Because when one thinks about a tumor, we think, we tend to think about the cancer cells or the somatic mutations or the germline mutations, but in fact, there are a lot of things that are happening at the same time, and and they're also changing through time. So it's a really messy thing. So for example, very, very silly example. Imagine that you have this patient, they have a tumor, and in this tumor you have cells with a somatic mutation that lead to overactive EGFR, increased signaling through RAS, and then cancer cell growth. And you have this cancer cell growing here. But at the same time, the same patient might have also a germline variant, which leads to higher PD-1 expression, inhibition through PD-1, and inactive CD8 T cells. And these 2 things are happening at the same time and cross-talking with each other. And these are going to change. This combination might, in fact, just be unique to this patient. So unless we start to, we can try to dissect these things and study them separately. But at some point, we're going to need to embrace this complexity and start to study all of these things together at once, because otherwise, we're not going to make sense of this mess. And to do that, we are working on the different stages of this. For instance, the lab started by studying which somatic mutations matter, right? So this is one of my TCGA papers, where we did the sort of the catalog of cancer driver genes. We analyzed, I think it was 10,000 patients at the time, with a lot of bioinformatic tools. I think something like 20 or 30 or something like that. And in the end, we integrated all that information to a final list of 258 genes. 87 of them can be found in more than one cancer type. And this is a nice resource that it's important, but what I really like about this paper is not the list of genes themselves, but actually the list of mutations that we created. Because not all mutations in cancer genes or driver genes are actually oncogenic, right? You might have a mutation that is a passenger even in p53. So how can we distinguish those, the true cancer mutations in cancer genes from the benign ones? And to do that, we created a list of, let's say, 3 categories of mutation prediction algorithms. First, you have the classic ones, right? The regular tools to predict disease mutations using just the linear sequence of the protein. Here you can find SIF, polyphen 2, et cetera, et cetera. Then there are tools that also use driver mutations, linear features, but these are specific for cancer. And here you have CASM, CANDRA, FATE-HMM, or TRANSCRIPT. And finally, there's a third group of tools which are based on the spatial, in 3D, distribution of mutations across the protein structure. And here you have HotMaps, eDriver 3D, HotCode 3D, or other tools. And based on this idea, we identified, we were able to classify the mutations in each cancer gene according to, first of all, to the tissue where they happened, and into how many of these three flags were labeling the mutation as important. And here, this is a very nice result, I think. So here you have, for instance, let's focus on EGFR for a second. Okay, and here you have a column that says canonical and a column that says non-canonical. What do we mean by that? So canonical are the mutations that happen in cancer types where EGFR, we detect it as a cancer driver gene. This is mostly, in the case of EGFR, this is glioblastoma, lower grade glioma, and lung adenocarcinoma, right? But then there are patients with, I don't know, breast cancer or prostate cancer that also have EGFR mutations. But these are extremely rare and very, and usually, they don't happen in the hotspots. So these, so the canonical ones would be the ones in brain and lung, and the non-canonical ones would be any mutation in EGFR in any of the other cancer types. And you can see here very clearly that the mutations that the tools are predicting as really oncogenic, right? The ones that we can missense mutations, we predict with at least two of these three tools that they are oncogenic, they tend to happen in the cancer types where we know that this gene is relevant. Whereas the mutations that we predict to be passenger are happening mostly in these weird one-off mutations in breast or, I don't know, or thyroid or whatever. And this is true not only for EGFR, but also for ERBB4, GNAS, GNAC, and essentially the vast majority of cancer genes. These weird one-off mutations, which are likely passengers, they're happening in these rare cancer types. Not only that, but then we also wondered whether the three categories differed in their ability to predict these true cancer mutations. And here you have the three tools, right? And we were lucky and we were able to test them in actual cancer cell lines through CRISPR. And these are the validation rates. And you can see that the methods that were the best are the ones based on structure. So yay, now we have AlphaFold, so we can extend this to the entire human proteome. So this was another important result. And in the end, we ended up with a list, I think, is of 3,400 mutations that we really believe are the true oncogenic driver events that happened, at least in the TCGA samples. So now that we know which of these mutations are really important, the next obvious question is, okay, well, what do these mutations do? And here we are 4 years later. This is unpublished data. It's the first time that I'm presenting it, so I'm very excited. So this is the CPTAC project. The CPTAC is TCGA 10 times smaller, so it's not 10,000 samples, it's only 1,000. But now we have proteomics. So we've been working on this for the last year. It's under review in a very nice journal. And we have, for these 1,000 patients, we have a combination of somatic mutations, RNA-seq, proteome, phosphoproteome, clinical data. And for some, we even have like acetylome and whatnot, but we didn't use it here. And here this is, by the way, this is led by Kathleen in my lab. And here we did the simplest control that we can think of. Okay, let's start with the disease effect. What happens to P53, for example, when P53 is somatically mutated, right? What happens to its protein level? Do they change? Do they stay the same? And you can see here that, for instance, so here you have each dot is a tumor sample. And here you have the wild type, the patients without P53 mutations. These are the P53 protein levels and the P53 phosphorylation levels. And you can see that all the patients with missense mutations, they have higher protein and phosphoprotein levels of P53. The same is true for in-frame insertions and deletion. But then when you go to nonsense and frame shift indels, the protein levels are lower, which we expect because of NMG decay, and the phosphoprotein levels stay more or less the same. Okay, so this was like a control that the data makes sense. It all looks good. Let's make some more sophisticated things. So there's this long-standing hypothesis that if two proteins interact with each other, their protein levels tend to correlate, okay? Whereas if they don't interact with each other, their protein levels do not correlate at all. And we said, okay, we didn't measure protein-protein interactions in CPTAC, but we could try to use these as a proxy and see what happens, right? So first of all, does it work? And the answer is yes. So here you have, this is 25 million correlations. So we measured 5,000 proteins in CPTAC, so it's all possible protein-protein pairs. And these are classified based on the number of databases where the protein-protein pair is described as interacting with each other, okay? So 0 are proteins that do not interact with each other. And 3 or more would be the proteins that are predicted or that are described as interacting with each other in 3 databases or more. And here's the Pearson R value of the correlation. So when the proteins do not interact with each other, the average Pearson coefficient is 0, which is what you would expect by chance, right? Whereas in the proteins that we know very well that they are really interacting with each other, the Pearson correlation coefficient is way higher. And it tends to, it's biased towards the actual correlations. And here's a couple examples. I think this is in breast cancer. So MSH2 and MSH6, they interact with each other. They form a beautiful complex. Here's this awesome correlation. Beta-catenin and CDH1, same thing. They interact with each other. They form this awesome correlation. So the concept works, okay? So we then extended these to try to see cancer type and mutation-specific network rewiring. What do we mean by that? You have, for example, mTOR. mTOR can interact with Raptor or with Richter, right, to form the mTOR1 and the mTOR2 complexes. Don't ask me which is which because I always forget. But so here you have mTOR and Raptor correlating with, well, the correlation with each other, or the non-correlation in this case, and mTOR and Richter and the correlation. And here you have it in kidney cancer, okay? So we can see using the protein levels, this suggests that mTOR is signaling through Richter in kidney cancer and not through Raptor, okay? Okay, what happens in glioblastoma? Well, when we go to glioblastoma, the mTOR and Richter correlation is way weaker, okay? It's still there, but, this is really weak. ER is really poor. Whereas the mTOR and Raptor correlation now is suddenly very clear. So this suggests that in glioblastoma, it's not through Richter that mTOR is signaling, but it's actually through Raptor, okay? So we can start to see this cancer type specific rewiring of the protein-protein interaction network. And we can go one step further and say, well, what happens when you have mutations, right? So p53 can be degraded or targeted for degradation with UB3A through, ubiquitination. When there is no p53 mutation, you can see that very clearly because the higher the UB3A levels, the lower the p53. Okay, so it's negative correlation. It suggests that, UB3A is signaling p53 for degradation. But when you have a p53 mutation, this correlation disappears, which could mean, again, we don't have direct evidence for that, but suggests that one of the oncogenic mechanisms for missense mutations in p53 could be, that it's no longer targeted for degradation by UB3A. And then we can, when we take that into a more global picture, oh, these are not, I thought that the dots would be there, sorry. This is for a different paper. It focused more on glioblastoma, where we had also recurrences of the tumor in the same patient. And here you can start to see the patterns like, I don't know, this is the EGFR, the EGFRA, an entire signaling pathway all the way until RB1 and SMARCA4, right? And you can start to see interactions that are described in databases, but that we cannot see, which are the ones in dashed gray lines. Interactions that we detect in no matter what, no matter the mutations, no matter the time point, et cetera, which are marked in black, right? And then interactions that could potentially be affected by somatic mutations, like EGFR, for instance, in this case, we have it, I don't know. Yeah, I don't know, like PTPN11 and IRS1, which is PTPN11, IRS1, you have them here. This interaction might be affected by PTPN11 somatic mutations. Or even, since now here we also have different time points, we identified some interaction or some proteins that no longer correlate. They correlated in the primary tumors, but they do not correlate with each other in the recurrent tumors, like in RB1 and CDK2. So you can start to explore how all this network is being rewired. Another cool experiment is this one. So we also use the proteomics and phosphoproteomics data to quantify how similar are two cancer genes. So what do we mean by that? Imagine that you have, I don't know, PIK3CA, right, and EGFR. And then what we did is we splitted the samples into samples with PIK3CA mutations and without them. And we quantified how all the proteins, all these 5,000 proteins that we measured, and all these, I think it's 2,000 phosphosites that we also measured, how all of them changed depending on the status of PIK3CA, right? And in the end, you have this mutation here, and then this protein increases, and these other proteins decrease. Okay. And now we repeat the same experiment, but now with EGFR. And we get to this pattern. These three proteins increase, this phosphosite increases, and these two proteins decrease, okay? So we turn this into some kind of score, which is essentially, this is the p-value of the fold change. With a positive sign is if the fold change is positive, and a negative sign if the fold change is negative, so if the protein decreases, okay? And then we can calculate the correlation between the two, a sort of a proxy into how similar these two things are, right? So if you see a nice correlation like in here, you would say, okay, these two driver genes, their trans effects look very similar because they are changing the same proteins in the same direction. Whereas if we see a different pattern, then then the two driver genes would be different. And here's we calculated this for all possible pairs of driver genes, and this is the overall driver similarity landscape. Okay, in the extremes, you have things like NFE2L2 and CAP1 and EGFR STK11, which are the two pairs that I will explain in more detail. So NFE2L2 and CAP1, this is the simpler example, so to speak. These two proteins, NFE2L2 is a transcription factor. CAP1 targets NFE2L2 for degradation. The mutations tend to happen in this interface right here, okay? And these are mutations that are mutually exclusive. So when a tumor has a mutation in CAP1, it doesn't have a mutation in NFE2L2, and the other way around. Okay. Here's the scores that I mentioned for CAP1, the scores for NFE2L2, and you see that they have this very nice positive correlation. Because in the end, no matter which of these two mutate, the end result is that NFE2L2 ends up being in the nucleus and ends up targeting all the proteins that are regulated by it for overexpression, which are these ones, AKR1C2, GC11, all of these, right? So in the end, no matter which of these two mutate, end result overexpression of these proteins, which explains, which is what we call mutual exclusivity due to convergence, right? Phenotypically, these mutations, they converge. They end up with the same result, overexpression of these proteins right here, okay? But then there are also these negative correlations, like EGFR and STK11. And here the story is a little bit different, because whatever is upregulated in EGFR mutated samples is downregulated in STK11 mutated samples, and the other way around. Upregulated in STK11, downregulated in EGFR mutated. So, and you see very clear here, PTPN11, Y62 phosphorylation, down in STK11 mutants, up in EGFR mutants, and IRS2 phosphorylation, up in STK11 mutants, down in EGFR mutants. And here, this is a hypothesis, and take it with lots of caution. But our idea here is that these mutations, they are also mutually exclusive, like NFE2L2 and CAP1. But in this case, it's not because they converge, because clearly they are overexpressing and overphosphorylating very different things. In fact, the opposite things. So what we think that's going on here, it's incompatible divergence. So we think that whenever you have an EGFR mutation, for example, it pushes the cell towards a certain state where an STK11 mutation essentially would kill it, because it would start to phosphorylate other things and trigger some pathways that would end up killing the cell. And this is why we don't see the commutation of EGFR and STK11. This has been shown, by the way, for EGFR and KRAS in lung cancer. They are synthetic lethals. And I think for, I have to check the paper again, but I think also for 53 and CDH1. So it kind of makes sense. Okay. This part, I think I'll skip for the sake of time. Just know that somatic and germline mutations are important for the immune response against tumors, and you can use them to predict it. Okay. Okay, and this is the part that I think fits most with the rest of the speakers. So spatial transcriptomics. I'm going to give a very quick overview so that the other people don't have to. We use Visium in the lab. Daniela uses Visium in the lab. I don't, because I would mess up the experiment. So Visium is based on these microscopy slides. They are very nice. They have 4 capture areas, 6.5 millimeters, 6.5 millimeters. And the nice thing about these capture areas and why this slide costs 3,000 euros is because it has these 5,000 barcoded spots, which are 55 microns in diameter. And from center to center, you have 100 microns, right? And then these spots are filled with oligos that they contain this poly-DT to bind the mRNA, the UMI, so that you can count which molecule the RNA is coming from, and the spatial barcode, so that you can then, once you sequence a library, you can map the read to the exact spot where it's coming from, okay? And using that, we can generate high-quality data. These are 2 serial sections of an ovarian tumor. So this is the first section, this is the second section. So the same tumor, same, these are 10 microns apart. And here you see the expression of MYC, and it's very clearly clustered in this area, which suggests that these are the cancer cells. And this is the same exact area in the 2 sequence slides. And TGF-beta, which suggests that this is the tumor microenvironment, right? And again, same exact location in the 2 serial sections. So, I think it's nice. Just know that we offer these as a service, and we are #1 in Spain officially. We processed over 160 samples, and I think it's like 20 different cancer types or tissue types. So if you want to do it with us, we have quite a lot of experience, and we can help you. And then with all of the samples, we published this preprint last June, charting the spatial landscape of cancer hallmarks, which is led by Mustafa, Sergi from the bioinformatics side, and by Daniela from the wet lab side, and a lot of other collaborators that helped us collect the samples and analyze them. Okay, and here the idea is that we wanted to study the cancer hallmarks, okay? And for those of you that don't know it, which is probably no one, but just in case, the hallmarks of cancer, where this was a concept, sort of a heuristic tool developed by Douglas Hanahan and Weinberg back in 2000, where they said, well, okay, if you look at cancer, right, it's all this complexity, but it can be, all tumors need to end up having, at that time it was 6 features. I don't remember which ones, okay? But now in this January, they published in Cancer Discovery the latest iteration of the hallmarks. And the idea here is that all tumors, no matter where they happen, they need to have all these 14 features, right? So they need to sustain proliferative signaling, evade growth suppressors. Usually they use non-mutational epigenetic reprogramming, and so on and so forth, okay? So these are like the features that every tumor needs to acquire. It doesn't tell you how. It can be through mutations, through upregulation, through, I don't know, different ways. But it needs to have all of these usually. And so we said, okay, can we use spatial transcriptomics to quantify where the different cancer hallmarks are happening? And here's, so we did this we created a series of cancer hallmark signatures it's not complicated, but it's, in the end, we ended up having like grouping like between 100 and 1,000 genes to each hallmark, okay, and creating these hallmark scores, which is the sum or the end result of these between 100 and 1,000 genes. It depends on the hallmark, okay? Just to show you that these hallmark scores work, here you have, and these are 7 of the 41 capture areas that we analyzed in the paper. You have the methoxyline in here. You have here a series of what we call marker genes, which are genes that people usually associate with different hallmarks, okay? And here you have the actual hallmark score. For instance, the hallmark evading growth suppressors. It has this pattern in this sample, okay? And here's a gene, CDK1, which usually people associate with evading growth suppressors. And you can see that the pattern is similar, but not quite the same, which is why we think it's important to have a cancer hallmark signature instead of an individual marker that would change from cancer type to cancer type, okay? And here you have the same for, I don't know, EZH2 and non-mutational epigenetics, BRCA1 and genome stability, and so on and so forth, okay? So this was like the first result that we got, and we realized that most hallmarks are expressed in very specific areas of the tumor, like here, EZH2, right? It's expressed only in this area, but not in these other areas. Or activating invasion of metastasis is active here, but not in this other area. So we wonder whether that, or our idea was that this was probably coming, you had hallmarks that were coming mostly from cancer cells, and other hallmarks that would be coming mostly from the tumor microenvironment. And it is the interplay between both that would trigger oncogenesis. But to test that, we first needed to find where these cancer cells are. Okay, we started doing this with pathologists. They have very nice results, and it's what's described in the paper. But now for the latest iteration of the paper, we are using Estimate. Estimate, for those of you that don't know it, it's a bioinformatic tool that it was used a lot in TCGA. The idea is that you can give it a bulk RNA-seq, and then it tells you how many cancer cells you have there, or how many TME cells you have. Okay, so if it's more cancer pure or cancer less pure. Yes. But then we said, okay, if this works in bulk RNA, these spots they usually have between 5 to 10 cells. So it's like a mini bulk RNA. So we use Estimate to calculate, or we calculated the Estimate score for all the individual spots in each Visium slide. Okay, and this is the end result. This is a colorectal cancer sample, I think. And this is the Estimate prediction. And you can see that whatever looks like a tumor, right? It has a super low Estimate score, which correlates with what Estimate is telling you that it's cancer, and the other way around. Whatever, you cannot see it here, but our pathologist told us that there were lymphocytes there, okay? So believe us. Whatever the pathologist told us that we had lymphocytes, it had very high Estimate scores, which correspond to the tumor microenvironment. So we can very easily use that to classify the tumor into what we call cancer pure areas and TME pure areas, okay? And here's the same concept but now classified, instead of having a continuous score, we need to binarize it because otherwise the analysis is a bit messy. So we ended up with these 5 categories, one being what we call the cancer pure, and 5 being the TME pure, okay? And you see that it overlaps with what I've just told you. And then we calculated the average hallmark score in each of these 5 areas. And you can see that the picture always looks the same. You have hallmarks that have super low activity in the cancer pure areas, like in this case, this is activating invasion and metastasis, and super high activity in the tumor microenvironment pure areas, and the other way around. Senescence in this case, for example, is super active in the cancer areas, and very low activity in the tumor microenvironment areas. Okay, so when you extend this, like we did in the paper, to 15 samples, you get into this clustering. Here, each of these columns, okay, corresponds. It's not exactly like that. If you read the paper, you'll notice, but the idea is the same. Corresponds to one of these sub areas in a Visium slide. Okay, so like to a cancer area or a TME area. And each of the rows corresponds to a cancer hallmark. And you see, first of all, there's this very nice clustering, okay? And this clustering suggests that there's a pan-cancer architecture. So here you have, in red, you have all the areas that we label as tumor in the samples, okay? And in gray, you have the tumor microenvironment. You see that there's this very nice cluster of tumors together, of tumor areas together, of cancer areas together, and this other clustering of tumor microenvironment areas together, okay? This clustering is not driven, so for those of you that don't know it, this was an important internal check. Visium comes in 2 flavors or for 2 platforms. You have the paraffin-embedded samples and the fresh frozen samples, which are based on OCT. And the technologies are a little bit different, okay? So here, just to be sure that this was not messing the experiment, we also check whether there was clustering by the embedding method, and there isn't. Okay, so I think in pink, just to be sure, yeah. So dark pink is paraffin, light pink is OCT, and there's no clustering by that either. And there's no clustering by tumor type. Here we had, I think it was at the time, yeah, I think it was like 7 cancer types and 15 samples, and there's no clustering by that either, okay? So and then the idea is that you have a series of what we call cancer compartment hallmarks, non-mutational epigenetic reprogramming, resisting cell death, evading growth suppressors, et cetera, et cetera. And then there are hallmarks that are coming from the tumor microenvironment area, the regulating cell energetics, tumor promoting inflammation, inducing angiogenesis, et cetera, et cetera. So we published this preprint. I talked to a couple editors to see if they liked it. And the most common feedback, and I mean, it was fair, was that with only 15 samples, you cannot claim that this is a pan-cancer architecture. So this was June 23rd. We spent all summer working really hard. And here's a hallmark with 41 samples from 9 cancer types. And this is now an old version because we have it with 56 samples and 10 cancer types, and it looks the same. So here you have the tumor microenvironment hallmarks, the cancer hallmarks, and the pattern now is even more clear. So I hope that the editors will now be a little bit more convinced, okay? And here you might argue, okay, but Edu, you don't need spatial transcriptomics to do that. Because technically you could just use single cell, okay? Disaggregate your tumor, label the cells into cancer and non-cancer, and then you quantify the hallmarks there. You don't need spatial for that. And that would be true. But what you cannot do with single cell is this. So here, actually, let me take it to the next slide because it will be easier to explain. Here, if you're a cancer cell, you can be in 2 situations. You can be a cancer cell or a cancer spot, because it's not single cell yet. You can be a cancer spot surrounded by other cancer spots, or you can be a cancer spot surrounded by tumor microenvironment. And the other way around for the tumor microenvironment. You can be a tumor microenvironment surrounded by cancer cells, or tumor microenvironment surrounded by tumor microenvironment. And to quantify this in a more systematic way, sorry for the typo, we try to measure the neighborhood cancerness of each spot. And this is like a simple, I mean, this was like the simplest approach that we could think of. We created a circle around each spot of 200 microns, because this is usually where paracrine signaling, most paracrine signaling happens. And it's just so a weighted sum, weighted by the inverse of the distance. So the closer the spot is to the spot that you're studying, the more weight it has. The farther away it is, the less weight it has, because we think that the closer you are, it's more relevant. Okay, and you end up with this score where if the score is very high, it means that you are surrounded by tumor microenvironment spots. And if the score is very low, it means that you're surrounded by other cancer spots, which is what you see here. Here you have these 2 spots. Okay, I don't remember which sample is this, but they have the same exact estimate score. So from the spot itself, it has the same cancer score. Okay, it looks from the thing in terms of cancer purity, it looks both spots look exactly the same, but they differ quite a lot in the neighborhood score. And in this case, what the neighborhood score is telling us is that this spot is a cancer spot surrounded by other cancer spots. And this spot is a cancer spot again, but surrounded by tumor microenvironment. So what we did next is, well, how much does this matter? And this is like in the bare minimum that you could get, okay? So at the very least, in the worst possible scenario for the neighborhood score, okay, what we did here is this is just a simple linear regression where the predicted value is the hallmark activity. So here you have the different hallmarks. Okay, so H1, H2, et cetera, et cetera. And this is predicted first by estimate, which we know from the heat map so whether the spot is cancer or TME, and we know from the heat map that is going to predict that is going to drive most of the signal. Okay, because there are the hallmarks come clearly from one of both sides. And then we added a second term, which is the neighborhood score of each spot. And then we ask, okay, how much can we predict using only estimate, right? And this is shown with these light blue lines. Okay, so the higher the line, the more we can predict. This is actually the R value of the regression. So the higher the blue line, the higher the R, the more we are predicting. And then on top of that, we added in dark brown, I would call it, how much when we add the neighborhood score, how much extra we are predicting on top of what we know from the cancer. Okay. And here you have the experiment. When you take the whole slide, by the way, each column here is a cancer type, so because otherwise we will get too many lines. So on the periphery, you have how much the neighborhood score adds in when you take the whole slide into account. Okay, so both the cancer and the TME. In the middle, you have how much it adds for the parts of the slide that we label as tumor microenvironment. So we exclude here all the cancer, and we say, okay, how much does it matter for the tumor microenvironment cells? And you can see here that the brown lines are much higher. And in fact, in some cases, they are even higher than estimated itself. So what this means is that for the tumor microenvironment areas, whether they are closer or farther away to the cancer cells, this affects a lot their hallmark activity. And in the bottom area, you have how much it adds to the cancer areas only. And here it's a little bit lower because I guess that once you're a cancer cell, it doesn't really matter what you're surrounded for, okay? And so the next direction that we want to take this into is what we call cancer hallmark activity maps, where you have, this is the breast cancer sample that we analyzed. And if you run it through this software, then you will get for each spot, which of the different hallmarks is the most relevant one. Like, I don't know, here, this green area is sustaining proliferative signaling, whereas this purple area is, I guess, this would be deregulating cellular energetics. And this red area would be inducing oncogenesis, so that you can see which part of the tumor is doing what. And just to finish, the whole idea, I hope that I've convinced you that we need to embrace complexity and study, the mutations and the proteomics and the spatial and all of these things together to understand how tumors work. And that's it. Thanks to the people from my lab, all the different collaborators, and the funding agencies, and to you for your attention.